And one thing we have to kind of be considerate of in regards to the system is we are really in tune with what our body actually needs in regards to the status of the cell, whether we are in a high oxidative or low oxidative state, whether we need ATP or not. And this is where basically the crux comes down into regards to this consumption and need for ATP and the consumption of glucose to give us that ATP. We're talking specifically the role of phosphorylation, how phosphorylation can have a positive and a negative effect depending on the enzyme it controls. And we saw specifically when we're looking at glycogen breakdown to allow us to produce glucose circulational component in the form of glucose 1-phosphate, which will go to glucose 6-phosphate ultimately, or we can actually remove that glucose 1-phosphate and bring glucose into the circulatory system to get elsewhere, to the brain, to the tissues, etc. But within the liver, we have this particular glycogen store and some in the muscle, where we're basically using this reversible phosphorylation to go through the activation or inhibitional process. So in this case, with the glycogen phosphorylase A, it is phosphorylated, and when you minus the phosphorylation, it becomes then less active. So it doesn't say it switches it off completely, it becomes less active. So again, it's one of these systems that the foot is off the accelerator, but you're just idling at the traffic lights. We then also looked at this issue regarding both the cascade effect of glucagon, the hormone, peptide hormone, and epinephrine, which is a neurotransmitter in regards to the fight and flight mechanism, where we're seeing ultimately this massive increase as a cascade moves through with more enzymes being active and more substrates being produced in regards to the number of molecules that are coming through, as we see with a simple stimulation of both these particular systems through the GCPR and the cyclic AMP concentrations going through all the different phosphorylases and kinases to basically degrade glycogen to give us the many thousands of molecules of glucose 1-phosphate, which can then be brought into the blood or go through produce glucose 6-phosphate through glycolysis to give you muscle contraction, that ATP requirement with the actin myosin heads, and that process as we already seen in the first part of the lecture series. We're talking about then also UDP glucose, this uridine diphosphate starting product in regards to utilizing the glycogen synthase to elongate the non-reducing chain or the non-reducing end of the glycogen molecule. So we should have a clear understanding on that, but this is where I want to get back to in regards to this process of phosphorylation. So in this case, we have the active glycogen kinase number 3, which is active, which is basically promoting the inhibition of the glycogen synthase. Now, remember we talked about the glycogen synthase needs a priming action, so that's another piece of regulatory control where we have to have a phosphorylation to occur on the substrate already to be recognized by the enzyme to actually do the work. If not, we don't get that interaction. So phosphorylation plays a key part in this case in both inactivation of the glycogen synthase kinase number 3 and also the inactivation of the glycogen synthase to prohibit glycogen synthesis. And we talked specifically about another enzyme that's playing a role in the glycogen synthase kinase activation with the PP1 enzyme. And again, illustration of how phosphorylation causes inactivation and dephosphorylation in this case, using the enzyme, the PP1, to allow us to actually remove the individual phosphates off that, and having all 3 of those phosphates removed from the serine moiety will then activate the glycogen synthase kinase number 3. We have one issue in regards to bringing this back to the non-active form or the inactive form, is that we need that kinase caseinase kinase number 2 or casein kinase number 2 in regards to having that priming reaction. So that's not a part of the consideration of the amount of ATP normally taken to take these three moieties to become phosphorylated. This is an additional separate component, which then allows us to actually then move the molecule to increase its affinity with that priming component, as we see here in regards to the casein kinase number 2 doing that priming component and allowing then the glycogen synthase kinase number 3. So again, an illustration of regulation. But again, this part of regulation can also be instigated in auto or pseudo regulation in regards to you pretending to be a pseudo substrate inhibiting the glycogen kinase number 3. So that's where we see this guy here, the glycogen synthase kinase number 3 phosphorylated being inhibited in here. Okay, so phosphorylation gives us that inactivity. And then obviously we have our role of a GCPRs and also in this case the insulin receptor and insulin substrate receptor number 1 complex playing an important role in the protein kinase number B and going through the process of phosphorylating that to then get this guy to actually go through the inactive form to be inactive, and then the PP1 to then remove those phosphate groups to become then the active glycogen synthase glycogen synthase kinase number A. Sorry. But what I'm trying to indicate here that when we look at some of our enzymes, there are multiple complexes that are forming. These enzymes are not by themselves, whether they are cytosolic or membrane bound. They're going to attract other constituents, which will form a complex, as we start seeing here with the PP1, which is central to this glycogen metabolism. So this is going to integrate various kinases and synthases and phosphorylases, and they're going to come together as a complex of proteins. Without one of those particular parts, we may not have the process of regulation or control. And we see here how insulin and epinephrine have different impacts on those particular association of proteins. And again, the PP1 having these particular roles as well as other inhibitors playing a role. So you can start seeing a weave of complexity occurring when we take a simple viewpoint of an enzyme substrate interaction. There are many other interactions, modifications, allosteric modulations, and regulatory modifications that are occurring in regards to this enzyme coordination. So we have this whole system in regards to the dephosphorylation of the PP1, needing three basic enzymes to undertake the chores in regards to the phosphorylase kinase, the glycogen synthase, and as well as the glycogen phosphorylase. So with this particular process, we're going to have multiple points of regulation and control, regulation upon regulation. So in this case, the glycogen targeting protein, in this case, what we call a GM, actually has two different sites for activation. And this could be either one for an epinephrine or insulin. So again, different sites, different points of regulation. So you can start seeing and kind of understanding the complexity that occurs in this central mechanism. Because we want to ensure that really we are fine-tuning our response and to our needs of the cell. Again, this switch between glycolysis and gluconeogenesis is our process of survival and how we've evolved as multicellular organisms to basically utilize our resources efficiently and effectively. So I want to really focus on this. This has sometimes a tendency to pop up on the exam in regards to understanding what's going on in the system altogether. And you should be able to start thinking about this in this particular way in regards to you have a very high glucose intake, i.e. your blood glucose is high. In our first response, we're going to see the insulin secretion from the pancreas increase, because that is going to try and move the high blood glucose concentration into the cells and lower the blood glucose level. And we need to do that by first increasing our transporters. So the GLUT2 transporters are going to be activated, and they're going to increase the concentration inside the cytosol of the cell, lowering ideally the blood glucose level. But as we see, we need to have that coordinated with our insulin activation, which is going to have a whole load of consequences in regards to activating various enzymes and systems to either promote the degradation of glycogen through glycolysis or push that glucose in the form of glucose 1-phosphate to make basically glycogen through the UDP glucose system. So let's have a look what occurs when we see that insulin level. Through our pyruvate kinase B, we're going to see the activation, in this case, the decrease in the glycogen synthase kinase number 3, therefore promoting the avenue to allow us to have the activated glycogen synthase. So we're taking up the glucose and going through the process and increasing glycogen synthesis. We also do that in parallel with the insulin-sensitive protein kinase as well as the PP1 in regards to its activity to activate the inactive form of the glycogen synthase. And in this case, we're going to see a decrease in the glycogen phosphorylase, which is the one that's going to be basically breaking down the glucose, or the glycogen. So in this case, we're promoting glycogen synthesis and glycogen storage at the cost of the glycogen phosphorylase. So we're basically prohibiting the breakdown of glycogen because we want to form more of it. We're going to also require phosphorylase enzymes to help in that help inhibit that breakdown. So we're going to have the phosphorylases also inhibited in regards to also decreasing the glycogen breakdown. In the same process with the insulin, we're going to see the hexokinase playing a particular role, the phosphofructokinase number 1, the pyruvate kinases, all doing their particular job to promote a clear pathway to allow us to sink the glucose in to increase the ATP and the NADH concentrations by basically promoting glycolysis. And again, that is going to be with that phosphofructokinase number 1 at the expense of gluconeogenesis. So again, we're shutting down gluconeogenesis, we're increasing glycolysis, we're increasing the movement of glucose into the system, we're decreasing the capacity to break down glycogen, we're increasing the capacity to increase our glycogen synthesis. So again, in response to and hopefully lowering that high blood glucose level. So again, this is what we call a well-fed state. So just think about your own bodies in regards to your high glucose. You're going to do something with it. You have to get into the cell, you're going to promote its utilization as glycolysis, or you're going to promote its utilization as a storage moiety in regards to glycogen. Now in the fasting state, there's basically a switch of different systems to allow us ultimately to combat a decrease in blood glucose where it's rapid, but also importantly allow us to hopefully come back to homeostasis. In this case, with the lower blood glucose, we're going to see the hormonal peptide glucagon being increased by concentration. We're going to see it interacting with this GCPR to increase cyclic AMP and also promote the protein kinase A activity, which is going to also have a role in our switching part. So here's our fructose bisphosphatase 2 and our phosphofructokinase number 2. So we're going to switch these two by having the protein kinase go through the phosphorylation event in regards to increasing the protein, the fructose bisphosphatase number 2 at the expense of the phosphofructokinase number 2. So again, promoting the issue of gluconeogenesis at the cost of glycolysis. So in this case, we're going to see a decrease in the fructose 2,6-bisphosphate and then a decrease in activity in the phosphofructokinase number 1, which is then going to then basically favor the process of gluconeogenesis and also prohibit the process of glycolysis. So again, we see a decrease in glycolysis, we should see an increase, perceptible increase in gluconeogenesis. We also see that through inhibiting our substrate going to the TCA cycle through the pyruvate kinase and basically shunting that glucose off to glycolysis. So again, we're stopping that avenue and then pumping everything back up to the production of gluconeogenesis at the cost of glycolysis. So we're decreasing the glycolysis level. In the same process with the glucagon receptor, we're going to also decrease the glycogen synthase. So get that glycogen synthase phosphorylated, decrease the activity, and then prohibit further production of glycogen, which we're doing here by basically interfering with the enzyme via phosphorylation. In the same process, using the protein kinase A, we're going to increase the phosphorylase kinase activity. That is going to give us then the glycogen phosphorylase capacity to bring in those darn enzymes to start breaking down the glycogen to give us glucose 1-phosphate, ultimately getting to the mutase reaction to get glucose 6-phosphate, and then feed that down into the pathway of distributing that glucose. This is mainly in the liver, but distributing that glucose specifically into circulation for the brain. So again, we're increasing glycogen breakdown, releasing those sugars. We're not putting those sugars through gluconeogenesis because they're already a commodity, and we're going to have those circulated as a rapid response, mostly for a very serious decrease in sugar levels and trying to maintain, importantly, homeostasis. Now, we also know from the MAPK cascade that when insulin is secreted and works on its particular receptor and involving the phosphorylation of the insulin receptor substrate number 1 and the GERB and the MEK and the MAPK and everything else, we are going to see other enzymes also being expressed. And these enzymes are correlated to the issue of fat and carbohydrate metabolism. Because obviously, if we are stimulating the system to bring in glucose, we're going to have to make sure that our enzymes are functional and that we can express more of these enzymes that are needed. And because we're in a starving state, we might go and utilize our glycogen and our glucose stores, but when they are depleted, we're going to have to make sure that we have something to substitute that. And that is going to be through the issue of fat metabolism. We'll look at fat metabolism in about a week's time. And we're going to be basically looking at how we coordinate this particular approach in beta oxidation to give us acetyl-CoA, which is going to be then a primary component for metabolism and maintaining our energy sources, though it's not preferential in regards to it's more longer, more energy inefficient, and basically can cause some other issues and problems, particularly in acid-base balances. And we'll have a look at those a little later on. So when we see insulin being bound to the receptor, just don't think it's regulating the enzymes for glycolysis, for gluconeogenesis and glycogen synthesis. It also is activating other enzymes, including those that are interacting with fat metabolism. So as you can see, there's a whole feature of enzymes, that some of these we've already come across, in regards to the issue of regulation of insulin. A lot of these are with glycolysis. We have one, the seesaw with the phosphofructic kinase 2 and the fructose bisphosphatase number 2, which is that key point in regulation of glycolysis of gluconeogenesis. We have the pentose phosphate pathway involved there. These guys we haven't come to yet in regards to fatty acid synthesis, but also with the triacylglycerol synthesis. So again, trying to promote the utilization of this energy storm, and if it's in the surplus, move it to something else that we can have a longer term storage component in regards to hopefully maintaining our glycolysis or our glucose level. Okay, so we come to really then the summary of what we've seen before in regards to the movement of the GLUT4 transporter as well as the GLUT2 transporter, in regards to the insulin receptor playing a very important role in the transporter as well as the commitment stage for glycolysis to go into glucose to go into glycolysis or for glucose to go into glycogen storage. So again, insulin is playing a very important role, as well as that glucose 1-phosphate, which has the phosphoramidase reaction, so we can shift that from the 1 to the 6 position. But again, ultimately, it is all to do with the issue of the insulin substrate receptor and the protein kinase B playing that phosphorylational role, indicating the activation or deactivation of the glycogen synthase, as well as coordinating with the transporters to bring the carbohydrate into the system to meet the demands of the cell. So I want you to look at the bypass reactions in glycolysis and gluconeogenesis, the role of the fructose 2,6-bisphosphatase and its regulatory component in the seesaw of glycolysis and gluconeogenesis. And we take a closer look of glucagon and insulin, how they play a role in maintaining and fluxing that issue in regards to response to the glucose concentrations and coordinating that in regards to glycogen synthesis and breakdown. So again, when the cell needs it, when the cell doesn't need it, and when the cell is happy in regards to an active or a resting state, particularly as we see in muscles. We also think about the issue of hormonal and allosteric regulation in regards to the liver, the cyclic AMP cascade, where both the epinephrine and the the insulin has a major impact in cascading and increasing the number of glucose molecules either being committed to glycolysis or to moving those glucose molecules, particularly in the liver, out into circulation to actually benefit the rest of the body. We look at specifically the glycogen synthase A and B, the glycogen synthase kinase number 3 in its process of pseudo-auto regulation with that phosphorylation event, and as well as the PP1, which is central to that metabolic process of phosphorylation, dephosphorylation between the phosphorylase, the glycogen phosphorylases. And then I want you to really then focus on understanding what goes on with the well-fed and fasting state. And it should be kind of pretty logical what you now know in regards to, I need transporters, I need to put that glucose somewhere, I need to either metabolize that glucose or put it into storage. What am I going to switch on and which am I going to switch off in regards to basically streamlining that event to get where I need to be. So that kind of concludes the material on glycolysis and gluconeogenesis. But we're going to have continual coming back to this a little later on in regards to what's really going on with metabolism. So we're going to see importantly how now we move from glycolysis to the next stage of metabolism, which is going to be the TCA cycle. And we're going to specifically see how glucose modifies itself into the pyruvate, and pyruvate into then the first decarboxylation reaction to give off the carbon dioxide. And then we're going to ultimately see the acetyl-CoA playing the key component in regards to this particular reaction and this particular pathway. And you're going to have to know, whoops, that's the wrong one. You're going to have to know this particular pathway. Let me get this up. Okay, escape. So we finished lecture 27. And let me get on to lecture 28.